Assalamu alaikum. Very important practical questions with answers of your microbiology in third semester. See, counting of microorganisms, it's one of the very unique and maybe in some colleges these question ask for major and also minor. See, aim is the report the concentration of bacteria or fungi in a given sample by using the counting chamber. Now, how you have to count the microorganism means you just read this all theory and requirements principle. Just I will go through the procedure for this directly. Place a drop of a bacterial suspension made from liquid culture and let the preparation stand for 1 to 2 minutes to allow the microbes to settle. See, what you have to do means you have to take one newborn slide. In that newborn slide, see, in that newborn slide, what you have to do, you have to add here 1 to 2 drop of the culture what you have given, no, that bacterial suspension you have to add in that newborn slide. Then put the cover slip over the grin carefully so that no air bubble enters into the cover slip. You have to put the cover slip here so that no air bubble should be absorbed in that between the cover slip and the newborn slide. How you done your WBC and RBC, in the same way you have to conduct this experiment. Then what you have to do, count the number of cells in middle square which consists of the 25 loops of one square, and for larger pores count the pores of bacteria and four corner larger square, and in the middle on the middle one to have total count of bacteria. Very simple. How you counted RBC, WBC in the newborn slide, in the same way you have to count here. That's simple. And report, see this report you have to write it down. Next, simple staining. Simple staining, it is also a major and sometimes minor experiment. This in here you have to write aim they will give. Principle is mandatory for all. And in synopsis you have to read all the principles. All the principles you have to cover of your microbiology practicals. Then see procedure. Take a clear glass slide, wash and dry it. First you have to take one glass slide, wash it and then dry it. Then sterilize the loop of flaming and draw a loopful of culture from the given stalk sample. First what you have to do means you have to take that glass slide, then wash that and dry. After that you take that glass and just rotate where on the flame so that the present microorganism should be dyed there. Then prepare a thin smear on the glass slide and dry the slide by heat fixing. Then what you have to do, you have to take that glass slide, you have to prepare a thin smear. See, this is the glass slide, thin smear. Just you have to take one microorganism with the help of the loop and just you have to prepare one thin smear here. That thin smear should be heat fixed. Then because if the microorganisms are alive means they should be dead in condition. Then about add about 2 to 3 drop any one of the given stain for the specific period of time. First you have to add that given stain. Then remove the excess stain by washing with the slow running tap water. Then dry the slide and observe under 45 into 10x objective lens. Simple. In simple staining what you have to do, just only one stain you have to write. Which stain they have given, like whichever may be, saffron, carbonyl, which stain they will give, you just add the stain, excess stain you have to wash with the running tap water slowly. Then leave for 2 minutes, just observe in the 45 into 10x objective lens. Then you write what the observation you observe there. Then organism blue means you observe there what is the color of the organism. Then shape cocky, okay, like bacterial classification based on the shape, you read that, you get easy. Number of organisms you observe there, how many number of organisms are there, you can write it. Means by guess you can write this all. Blue color cocky shaped microorganism submitted, studied and confirmed. Then gram staining, it is also very important one. Principle you have to go through that principle. Just we make a overview on the procedure. Make a thin smear of a bacterial culture on separate glass slide and fix the bacterial smell by drying. Same what bacterial culture they are giving, prepare a smear, thin smear. Then cover the smear with the crystal violet for 30 seconds and remove the excess stain by addition of the distilled water. Okay, first what you have to do, after getting that smear, just add a crystal violet indicator. Okay, leave for 30 seconds, then wash excess with the water. After that you again add grams iodine solution. Then add 95% of alcohol drop by drop for a few seconds and remove the excess alcohol by addition of the water. First you have to add crystal violet, then wait for 30 seconds or 1 minute hardly, then wash slowly with water, then add grams iodine, then again you add few drop by drop of 95% alcohol. Okay, then again you have to wash, excess alcohol should be washed by water slowly. Then add a saffron into the bacterial smear, dry the slide and observe under 45 into 100x objective lens, means 10x objective lens, or 100x or 45x. First this is the last process, saffron in addition, it is pink in color. There you will get the gram staining techniques, color purple, rod shaped, then number of organisms are moderate. See, moderate, you remember this one, don't write the numbers there, you write moderate, means if the organisms are more means you write rich, if they are if you are not getting any idea means you write the moderate. This is the technique, observation, gram staining. With the help of the staining we can observe the bacteria. Then mortality of the organism, it is also one of the major experiment. See, is to study the movement of given sample of organism. Major they will give a gram staining only, 100%. This one is a mortality of the organisms. Here in mortality, one thing you have to remember, don't heat fix, means don't ever go for the heating the smear. Okay, given sample smear you have to not heat, because here mortality should be important. You have to keep mortality forward, means the organism should be alive here, then only it will show motile nature. See, clean and flame hanging drop slide. Here flame hanging drop slide, that is the cover slide. Place it on the table with the depression upper side. You have to keep that depression where? On the upper side, like this way. Now this is the depression upper side. After that spread a little petroleum jelly around the cover, around the cavity of the slide. You have to take one cavity of the slide, cavity slide, then you add 1 drop, 2 drop, 3 drop, 4 drop, 4 corners petroleum jelly like Vaseline. Then transfer 1 loopful of culture in the center of the cover slip. Cover slip means now this is the cover slip, you have to add here only 1 drop of the sample what they have given, no liquid sample. Then place the depression slide on the cover slip with the cavity. You have to take that means that which one, hanging drop slide, that should be placed here, okay, on the petroleum jelly applied, and a drop of the sample containing cover slide you have to place on that. Okay, that drop should be covered by this which one, that hole. Then press the slide gently to form a seal between cover slip and the slide. You have to press down that one. Then lift the preparation quickly and turn the handling broth preparation covering up so that the culture drop is suspended. After dipping that, you have to, what you have to do means just lift that and just observe under the microscope immediately. Then examine the preparation under the low power objective, means 10x. Then with reduce light and place a drop of oil. This you don't take any confusion, just you remember until 7 points. Okay, just hanging drop, that experiment is only based on that. The main important thing is just by placing that drop inside that cavity, that is only main here. After that you just go and observe through the microscope. Better to you observe under the 10x microscope or 45x. 10x is best. Okay, then the given sample of microorganisms are motile in nature. It depends on your, if it is a motile, you write motile, means the tail will be moving of the tail there. If it won't move means you write non-motile. Next, just fermentation of carbohydrates. Then the antibiotic assay, it is also very important. See, very simple. Prepare nutrient agar plates containing 20% of 18 to 24 hours old culture. They will prepare you, don't worry. Then they are prepared by addition of 20 ml of microbial culture. Then, but before pouring media to the Petri plates, leave for solidification. After, make holes of 18 mm diameter with the help of the sterile cork bearer, and label the host and add respective drugs and incubate. Here this experiment is lengthy, it takes 2 days. So this experiment can't given, they maybe skip this one. Okay, which one? This one, antibiotic assay. You know antibiotic assay, you have done the assay for the antibiotics streptomycin in the examination. You can write that procedure. sometimes he will ask in the synopsis, write the procedure of the antibiotic assay. See, these are the very very important practical questions and answers for your examination. And synopsis, I have told you all the principles, 2022 question paper. In that you greater you give preference for the 2 marks questions. Okay, they will arise 2 marks questions in your synopsis there. And practical record is mandatory. Viva question they will ask in between this lab only. Practical labs, minor, major experiment, everything is completed. Okay, if you like this video, subscribe my channel Let's Join Pharmacy. And if you want more this type of videos, means on the 3rd semester B Pharmacy, remaining 3 subject, means comment down me in the comment section. Thank you. Assalamu alaikum.